Hello students, today I will talk about purification of immunoglobulin. Introduction. Immunoglobulins are very versatile molecules. They have been used in various immunotechniques like immunodiffusion, immunoelectrophoresis, ELISA, agglutination assays, etc. Some of these techniques use immunoglobulins without purifications like serum. In some of the specialized techniques like Western blotting, ELISA, immunofluorescence, etc., purified immunoglobulins are used. Purified immunoglobulins are further used for therapeutic purposes. They can also be tagged with various reporter agents such as enzymes, fluorescent dyes, gold particles, rare earth lanthanides, etc. for various applications. Purification of immunoglobulin. Purification of immunoglobulins involves many steps. In the first step, antibody samples are first characterized to check the antibody content by titering the antibody samples. Radial immunodiffusion and ELISA are some of the simple techniques to titer the functional antibody content in a given sample. In step 2, antibody clarifications are done to remove various other unwanted contaminants like cell debris, serum proteins, albumin, transferrin, lipoproteins, lipid-based impurities, etc. in the samples. The type of contaminants differs according to the type of antibody source. Antibody clarification is usually done by centrifugation and filtration. Other small contaminants and media components like phenol red, etc. can be cleared by simple buffer exchange using centrifugal filtration units or dialysis tubing. In step 3, precipitation. It is an important step to enrich the antibodies in the starting material. It can be of two types, positive and negative precipitation. In positive precipitation, the antibodies are precipitated, and in the negative precipitation, all the other proteins in the samples are precipitated. The methods for precipitation of immunoglobulin differs according to the source of the immunoglobulins. Some of the common precipitating agents are caprylic acid, ammonium sulfate, etc. Immunoglobulins can be purified from serum by adding chiotropic ions like saturated ammonium sulfate. At 60% saturation, ammonium sulfate can preferably precipitate immunoglobulins in serum. The precipitate fraction containing the immunoglobulins can be separated. In case of tissue culture supernatant, some of the media components like ferritin will co-precipitate along with immunoglobulin in ammonium sulfate solution. Therefore, further purification is needed. The best method of choice for further purification of immunoglobulins is affinity chromatography. Step 4. Affinity chromatography. In affinity chromatography, immunoglobulins are purified on the basis of their reversible interaction to a ligand present on the column matrix. The interaction between the immunoglobulin and the ligand are due to the formation of weak non-covalent bonds like electrostatic bonds, van der Waals forces, hydrophobic bonds, and hydrogen bonds, which could be easily formed and broken. These interactions can then be broken by adding some competing ligands in the eluent buffer, denaturing agents, or simply by changing the pH, ionic strength, or polarity. Immunoglobulin samples are passed through a column containing a specific ligand. Immunoglobulins will bind to the ligand and retain in the column. A washing step will remove all the unwanted impurities. Then an eluent buffer will dissociate the immunoglobulin from the column matrix and it is collected. Affinity purification is very specific and efficient. A single pass of samples in affinity column will give more than 1000-fold increase in purity of immunoglobulins. Affinity purification using protein A, G and L. Protein A, G and L are bacterial proteins derived from Staphylococcus aureus, group Z Streptococcus and Peptostreptococcus magnus respectively, and they are widely used for affinity purification of antibodies. One of the most widely used affinity column for immunoglobulin purification is protein A Sapphiros column. Protein A is a cell wall protein isolated from Staphylococcus aureus which has high affinity for immunoglobulins Fc region. It is a single polypeptide protein with molecular weight of about 46,762 Dalton. It has characteristics that are best suited to be ligands for affinity purification. Protein A is well characterized and can be produced in large amount using recombinant bacteria. It has molar dissociation coefficient of about 10⁻⁻⁷, which explains for their high affinity to Fc region of immunoglobulins. Protein A has high stability over a wide range of pH from pH 2 to pH 11. It can also sustain treatment with denaturing agents like urea and guanidinium and refold when the denaturing agents are removed. Protein A also have remarkable resistance to 0.5 molar sodium hydroxide, and therefore cleanup of protein A could be done without any damage to the ligand. Therefore, protein A has vast application in purification of monoclonal antibodies and polyclonal antibodies from serum samples, cell culture supernatants, bioreactor supernatants, acid fluids, etc. Immunoglobulin binds to protein A Sapphiros column at pH 8 and it is eluted at a lower acidic pH. One limitation of protein A affinity column is the use of eluent buffer with low pH, which can change immunoglobulin conformations. Therefore, attempts are made to produce recombinant protein A with lower affinity to immunoglobulins, which can be eluted at higher pH of about 4 and above. Protein G also has affinity for Fc region of immunoglobulins. However, it also has affinity for albumin, alpha-2 microglobulin and kininogen, which precipitates along with the immunoglobulin. This reduces the purification efficiency of protein G. Protein G also has less binding capacity compared to protein A, and therefore protein A are more preferred than protein G. However, protein A has restricted binding to immunoglobulins of only few species. Nowadays, many bioengineered protein A and G are produced and commercially available, which has broader binding capacity and efficiency. Protein L has affinity for kappa light chain of immunoglobulins. It binds to kappa 1, kappa 3 and kappa 4 subtypes but does not bind to kappa 2 and lambda light chains. Protein L has capacity to bind to various classes of immunoglobulins like IgG, IgD, IgE and IgM, and thus is used for purification of antibodies with kappa 1, kappa 3 and kappa 4 light chains, which constitute 50% of the human immunoglobulin. Affinity purification using antigens. Since antigens specifically binds to antibodies, antigens can be used as ligands to purify antibodies. Antigens are immobilized in the column matrix and act as ligands. Such affinity purification is useful in cases where antibodies with specific antigen specificity have to be separated from a mixture of antibodies with different specificity. It is also used for purification of antibodies with anti-antibody specificity. Affinity purification using anti-antibodies. Antibodies with specificity of the constant region domain of the heavy and the light chains could be used as ligands for purification of antibodies. Such anti-antibodies are embedded into agarose matrix and used as ligands. Such affinity columns are useful in purification of antibodies like red monoclonal antibodies which does not have affinity to protein A or protein G. Anti-red IgG are embedded in agarose and used for purification of red monoclonal antibodies. Binding is usually done at pH 8 and elution at a lower acidic pH. In such purification, cares are taken so that the acidic elution buffer does not damage the antibodies. Immediately after elution, concentrated buffers are added to neutralize the acidity. Since the antibody ligands are protein, care should be taken that these proteins are not degraded during the chromatographic process. Thiophilic affinity chromatography or TAC. It uses sulfur-containing ligands like mercaptoethanol in divinyl sulfone activated matrices which shows salt-promoted selective adsorption of proteins. Such matrices have high affinity for antibodies in the presence of high concentration of lyotropic salts. Lyotropic salts are those that can expose the hydrophobic region of proteins. Antibodies are then eluted from such columns by depleting the concentration of the lyotropic salts. TAC can be used to purify various classes of immunoglobulin. The main advantages of TAC are their use of milder elution buffer at neutral pH, autoclavable thiophilic gels, and low cost of adsorbents. One disadvantage of TAC is that the binding of antibodies to thiophilic columns are less specific than that of the protein A or protein G. But this has been exploited in purification of antibody fragments. TAC has been used in purification of recombinant FAB fragments without Fc regions and in purification of antibody fragments after papain digestion. Immobilized metal affinity chromatography or IMAC. Exposed amino acids like histidine, tryptophan, and cysteine on biomolecules have affinity for metal ions like nickel, copper, cobalt, zinc, iron, and gallium. In IMAC, metal ions are embedded in the column matrix which have affinity to proteins including immunoglobulins. IMAC is used in purification of recombinant antibodies where polyhistidine fusion tags of about 5 to 6 histidine residues are added which gives better binding efficiency. This histidine tag further facilitates purification of immunoglobulins with IMAC. IMAC is used in purification of vast range of antibodies including tumor-specific antibodies, single-chain fragment derived from monoclonal antibodies, etc. As a conclusion, I would like to say that purification of immunoglobulin is very important for the fact that it has varied applications. the best choice for antibody purification is affinity chromatography. There are different types of affinity purification depending on the types of ligand used. One of the most popular ligands used in affinity purification of antibody is protein A. Besides protein A, which has restricted affinity to only few types of antibodies and species, new ligands have also been developed for vast applicability. Some of these ligands are antigens, anti-antibodies, metal ions, sulfur-containing ligands, etc. Using these new ligands, the application of affinity purification for antibodies has been expanded enormously.